Welcome to AP Biology 7.1, Introduction to Natural Selection. I'm Gail Pfizer from Darnell Cookman, Jacksonville, Florida.
Hi, my name is Allison Kate. I teach AP Biology in California. Today, Ms. Pfizer and I will both be teaching parts of this lesson, but going forward, we will trade off with Ms. Pfizer teaching some lessons and me teaching others. Before Ms. Pfizer gives you the lesson overview for today, I wanted to provide you with the reasoning behind our approach to the online lesson planning. Today, the topic we are covering is one that colleges have asked all AP students to study, even if it will not be on the exam. Colleges are supporting AP students this year by allowing this more flexible approach to the exam, where it will focus on a lesser amount of content than in other years. But in turn, they have asked that AP students learn today's material. To make this relevant to your upcoming exam, we will also include some exam prep in today's lesson. For all lessons, it will be helpful to have pen and paper nearby, and for some of our lessons, we will request that you also have a calculator handy. In a week or so, after we've covered some of the key outstanding topics, we will be posting a full schedule of review topics for the remainder of this course so that you can choose which topics you want to prioritize for review and thus which classes you'll want to attend. We hope that you will attend all of the AP Biology lessons.
And now for a lesson overview. Today, we will be introducing you to the principles of natural selection and an overview of Science Practice Skill 2A, Visual Representations. You will gain proficiency in describing a given model or representation and communicating the biological meaning it represents. You should already be familiar with genetic variation and understand that genetic variation is necessary for natural selection, and using visual models and representations to explain or illustrate biological processes. By the end of this lesson, you will also be able to describe the causes of natural selection and explain how natural selection affects populations. And now we'll have a warm-up. Okay, so in the future, the warm-up will be the homework from the previous lesson. But tonight, we're going to start off with a skill 2A, the same skill that we'll be using tonight, but we're going to take it back to when it applied to cells. So I'm going to tell you how I teach my kids and how I go through either a multiple choice question and FRQs. So what I first tell them is to decide or to figure out what the diagram, graph, or table is telling them. It's going to tell them a story that's going to help them answer the question. So in this particular diagram, I would see 3 cells. There's a cell at 5 minutes, at 15 minutes, at 30 minutes. And I can see that it has a nucleus, an endoplasmic reticulum. It has a Golgi apparatus, secretory vesicles in each. And I can also see that these black dots are moving through this cell. And those black dots, I can see it's labeled figure 1, radioactively labeled polypeptides. So I can determine that there are black dots moving from the endoplasmic reticulum to, and if you look in the second cell at the 15 minutes, you can see one little tiny vesicle has a dark dot. So they're moving through the vesicles to the Golgi apparatus, and then the dark dots are leaving the Golgi apparatus and into another vesicle. Then that vesicle is fusing with that plasma membrane, and then the polypeptides are being released outside the cell. So I asked them to go through and get the story of what the diagram's telling them. And then I asked them to read the question so they'll know what they're going to be trying to answer. So the question is, using the experimental results, describe the pathway that secretory proteins take from their synthesis to their release from the cell. And then I asked them to read the passage if there's anything they think they still need after looking at the diagram. In an experiment investigating the mechanism of protein secretion, researchers tracked the movement of radioactively labeled polypeptides in pancreatic cells. At various times after the labeling, samples of the cells were observed using an electron microscope to determine the location of the radioactively labeled polypeptides. Figure 1 summarizes the results at 5 minutes, 15 minutes, and 30 minutes. The dark dots in the figure represent the radioactively labeled polypeptides. So I'm going to give you a minute to, on your pen and paper, with your pen and paper, write the answer to this FRQ question. Using the experimental results, describe the pathway that secretory proteins take from their synthesis to their release from the cell. And I'll give you just a minute to do that on your own. Okay, I hope that was enough time. Let's see what the scoring guideline says. And then you compare your answer with the scoring guideline. So the scoring guideline, you will get 1 point. This is a 1 point because there's one task verb involved, and I'll talk about those in a little bit. So if you said that it went from the endoplasmic reticulum to the Golgi, to the vesicle, to exocytosis, fusion with the plasma membrane, and outside the cell, you would have scored 1 point. Now, if you'll notice, there are words that are written in parentheses. Those are optional. Those are not required. Like if you did not say rough ER or rough endoplasmic reticulum, you would have not lost that point as long as you said endoplasmic reticulum. And the same goes with that second stage, which was the vesicle between the ER and the Golgi. So I hope that you scored that point. Okay, so the framework of the AP biology course. We've been working through different units this year: biomolecules, cells, photosynthesis, and we have broken those units into small topics. Each topic has had questions called the learning objectives. This is what we need to be able to do. This is what you need to be able to do on the exam. And we need to know content in order to answer these questions. So the content is called essential knowledge. So you need to know the essential knowledge to be able to answer the learning objective. And then each topic also has an associated science practice. And this is how we're going to show that we know the answer to the question, which is the learning objective. The science practice that is associated with natural selection is visual representation. You've probably been doing this in class since the beginning of the year. Okay, so Ms. Kate is now going to talk to you about what you need to know, the essential knowledge.
Hi, so I'm going to take you through the content to our topic today, which is an introduction to natural selection. And I thought a great place to start would be with a definition of natural selection. So I'm going to read it aloud, and I'd like for you guys to read it along with me. Natural selection is a process in which individuals that have certain inherited traits tend to survive and reproduce at higher rates than other individuals because of those traits. Now I'd like you to take a look at the graphics and notice that what we're going to be looking at is a prey-predator relationship. And there are 3 panels, and arrows moving from right to left are indicating time. So we're going to start at the first generation, and we see a population of mice has moved into a new area where the rocks are very dark. Due to natural genetic variation, some of the mice are black and others are tan. In the middle panel, we see that the tan mice are more visible to the predatory birds than the black mice. Thus, the tan mice are eaten at a higher frequency than the black mice. And since only surviving mice can reach reproductive age and leave offspring, in the final panel, we see that the black mice, because they had a higher chance of leaving offspring than the tan mice, the next generation contains a higher fraction of black mice than the previous generation. So just to take us back, this concept, this mechanism of how natural selection happens was first proposed by Charles Darwin in 1859. And if you look to the right, upper right of your screen, you're going to see what we affectionately call Darwin's finches. And when you look at these birds, you can see that these four birds all have very different beaks. They have different beak sizes and beak shapes. And so we're going to go through, using that as an example, the components that are needed for the mechanism of natural selection as proposed by Darwin. So the first thing is variation, and this exists naturally in populations, and I just went through the beak variations. We're going to talk more about how that variation gets introduced into populations in the next slide. The next component is reproduction. And the idea here is that more offspring will be produced than the environment can support. And that sets us up for the third component, which is competition. If there's more offspring than the environment can support, then there's competition for food, mates, nesting sites, and to be able to escape predators. The fourth component is the idea of fitness. In this case, fitness means that these individuals are more likely to survive and reproduce. So successful traits that certain individuals inherit mean that they are more likely to survive and reproduce. Finally, evolution occurs when these adaptations become more common in a population. So looking at the sources of variation in a population, the first source is mutations, which are random changes to DNA. The second source of variation in a population is due to sexual reproduction. When organisms reproduce sexually, there is a new arrangements of alleles in every offspring, and these new combinations of alleles mean new phenotypes. And the selective process works on the phenotypes, the traits that are visible. So again, this mechanism of natural selection was originally proposed by Charles Darwin in 1859. So let's put it all together. Let's look at the components that are necessary for natural selection, keeping in mind what Darwin's observations were and applying it to this scenario. So let's look at the starting population, and we're looking at, again, another prey-predator relationship, and we see that there's a snail species and it lives on a brown sandy beach, and the snails are eaten by hungry birds that spot them from above. There's overproduction, differential reproduction, and you can see that there are more juvenile snails that are born than can survive on this beach. There's not enough food or space for all of them to live. Some of the snails are always spotted by the birds and eaten. And as you can see, there's natural variation in this population. Not all the snails are brown. Some of the snails are red, some of them are green, some of them are blue. And so all of the snails on the beach are competing to stay hidden from the birds and not get eaten. Here's where that adaptation piece comes in. The brightly colored snails really stand out against that brown sandy beach, and that makes them easier to spot by the predators flying above. So the brown snails that match the color of their environment are more protected, making it easier for them to hide. The differential survival piece comes into play when, because of their color, the brown snails are better at avoiding the birds, and since they're eaten left, they can reproduce more. And in the final image, you can see that that brown sandy beach is mostly inhabited by brown snails. That means that they had a higher fitness and were able to survive and reproduce much better than the other variations in the population. And now what I'd like to do is introduce you to a real life example of natural selection. We're going to see the exact same thing that we saw in our previous example, but this is a real life example. I'm not sure if you're getting sound. I'm not hearing sound.
I'm not hearing sound either, but I can't, it's different.
Okay.
I didn't hear myself talk either.
Okay.
She can't hear it.
Okay,
in New Mexico, a battlefield, to find one of the tiniest souls.
Hold on.
Sand.
One moment, please.
there we go. That was,
Okay.
sorry guys.
Of volcanic
first
eruptions
time
that
through.
occurred about 1,000 years ago. The eruption spewed a river of lava more than 40 miles long across the desert. As the molten rock cooled, it darkened, leaving any creature dependent upon camouflage in serious trouble. In the complex battle of life, one of the constant struggles is between seeing and not being seen, the evolutionary game of hide and seek. And we've come here to the Valley of Fire in New Mexico, a battlefield, to find one of the tiniest soldiers and what it can teach us about how evolution works. On the desert sands, the rock pocket mouse blends in perfectly, its light-colored fur concealing it from predators. But on dark lava, the same fur makes the mouse stand out, attracting the many creatures that see it as food.
These mice are the Snickers bar of the desert. They're eaten by foxes and coyotes and rattlesnakes, and certainly by owls, and maybe even occasionally hawks. And most of those predators are visual predators.
You know, we haven't been. So what happened to the pocket mice that found themselves on this new terrain? When I accompany biologist Michael Nachman onto the lava, it doesn't take long to find out.
Oh, this one's closed.
Nachman has been collecting mice unharmed in traps. And it's a dark one.
It is, yeah.
Now, are most of the ones you find up here dark?
Almost all of them.
Not only have the mice here evolved to be as dark as the rock,
Shake them into the bag.
the color change has occurred precisely where it will conceal them from hunters. Now, a bit of a white underbelly, too.
That's right. All of the dark ones here and on other lava flows have a white underbelly, and presumably there's no selection for dark on the belly because predators are coming from above.
Left to themselves, the mice show no preference for light or dark rocks. It's the predators that have made the difference.
The change in color over evolutionary time in the population is driven by predators weeding out the mice that don't match their background.
But how did the dark mice arise in the first place?
And when a black mouse appears in a light population of mice, that is usually going to be due to a new mutation.
Sand.
Oops.
Okay, so that is the video clip just to give you a real life example of how protective coloration can be a protection from predation in that mouse population. So what I'd like to do is take you through a check for understanding, and this is the type of multiple choice question that you might have seen on the exam. So let's take a look at these side-by-side graphs, and the next slide will have 4 multiple choice questions, and you will need to select the correct answer. So when looking at the graphs, you want to make sure you look at the x-axis and see that it's showing you fur colors. You're going to look at the y-axis and it's showing you the frequency. And then in the right-hand graph, it's the same X and Y axes, but it's the fur color distribution after 20 years compared to the one on the left, which is the fur color distribution after immediate release. So let's take a look at the questions. We'll have an opportunity to come back and look at the graph before you give your final answer. So I'm going to read you the question. I'd like you to read it along with me, and then also take a look at the 4 answer choices. A group of mice was released into a large field to which no other mice had access. Immediately after the release, a representative sample of the mice was captured, and the fur color of each individual in the sample was observed and recorded. The mice were then returned to the field. After 20 years, another representative sample of the mice was captured, and the fur color of each individual in the sample was again recorded. Which of the following best explains the change in the frequency distribution of the fur color phenotypes in the mouse population, as shown in the figures above? So let's go back and take a look at those figures, and then you can come back and choose your answer.
Okay, can you describe that to them? Because
Okay,
I'm afraid if
so
I go backwards, I'm going to mess things up today.
okay, so we're unable to actually move backwards. So there were 3 color choices. There was gray, brown, and black. And in the first one, you could see that the gray bar was lower, and in this after 20 year, the gray bar was the highest. So hopefully that will help you with your answer choice. So let's read them. Choice A, the allele for gray fur color is unstable, and over 20 years, most of those alleles mutated to become alleles for black fur. Choice B, the field was composed primarily of light-colored soil and little vegetation, affording gray mice protection from predators. Choice C, sexual selection led to increased mating frequency of black and brown versus gray and brown. And choice D, the gray mice were the hardest to capture and so were underrepresented in the 20-year sample. So I hope that you selected choice B, that because there was primarily light-colored soil, the gray mice had protection from predators. And one final thing, while we're on the topic of natural selection, all of the components that I've been talking to you about were first described by Charles Darwin. There was another scientist by the name of Lamarck who also had a view of the mechanism of natural selection, and his view was called the inheritance of acquired characteristics. He said that physical changes that occurred during an organism's lifetime could be inherited by that organism's offspring. And the classic example is of giraffes and their long necks. So what Lamarck said was that as the giraffes stretched their necks during their lifetime to reach the leaves on tall trees, they would acquire those long necks. And when they had offspring, those offspring would be born with longer necks. While it's an interesting observation, it turns out that there is no data to support this. And I'll give you an example. So let's say you love Charles Darwin as I do, and you wanted to get a tattoo on your bicep. Then when you go to have children, your child will not be born with a tattoo of Charles Darwin on their arm. So that's the Lamarckian example.
Okay, on with the science skills. And today it's visual representations, and describe is the task verb that we're going to talk about. And describe characteristics of a biological concept, process, or model represented visually is the skill. And describe is a task verb. And we've used this particular one all throughout all the units of the curriculum, not just evolution. And when they're in a free response question, they're going to be bolded, and they're going to be worth 1 point on the exam. So when you describe something, this is all you need to do. You just need to list the characteristics, patterns, or trends of a representation. That representation could be a diagram, it could be a graph, it could be a table. And I'm going to, a lot of people get describe and explain confused. So I'm going to give you an example of each. If you describe mitosis, you could say, mitosis occurs in a sequential series of steps: prophase, metaphase, anaphase, and telophase. But if you were explaining mitosis, you could say something like this. You could say, during mitosis, sister chromatids separate during anaphase, ensuring the transfer of the genome to genetically identical daughter cells. So explain is more the how or the why of something. Okay, and I'm going to take you through a little guided practice on using this skill. So again, the first thing that I suggest you do, and I suggest to my kids, well, I kind of make them do it, but I suggest that you look at what the representation is telling you. And so let's look at the x-axis here. We have average beak depth of parents in millimeters. And on the Y, we have % of pairs successfully producing at least one offspring. So if I'm going to describe the trend illustrated, then all I would say is that I see a higher % of pairs successfully producing at least one offspring at 5 millimeters. Okay, let me read the passage just to see if there's anything else we need to know. Scientists investigated the role that beak depth plays in the ability of one species of seed-eating finch to reproduce. The scientists calculated the average beak depth of finches in mating pairs and then observed whether or not the pairs produced at least one offspring that survived to the next season. The data are represented in Figure 1. So what we need to answer is describe the trend illustrated. I'll give you just a moment because it kind of led you there for the most part. You should be able to write this down. Okay. So the answer, compare your answer, and hopefully you said something similar to this. Parental pairs with a specific beak depth of 5 had the highest reproductive success. Does yours look like that? I hope so. Okay, let's do another one as we're still under guided practice. I'll let you out on your own in just a little while. Okay, hemoglobin is the iron-containing protein used to transport oxygen in the blood. Since iron is important for growth, S. aureus have evolved the ability to absorb the iron from the host hemoglobin. Okay, I don't, now that I'm reading that, I go, I need to look at this table first. Okay, so at the table, S. aureus strain, I see 4 strains, 1, 2, 3, 4, and I see an amino acid sequence, and you'll see some letters that you might not be familiar with representing amino acids, like the Q is glutamine. You don't need to know that right now, but it can't be G because there's already another G. Anyway, host species, you have species A, species B, macaque, species C. And what we see in this table is that strain 1 prefers host A, and strain 2 prefers B, strain 3 prefers macaque, and strain 4 prefers species C. Okay, and what we're going to be looking for in the question, describe the process responsible for the different amino acid strains found on different hosts. So let's see if there's something in the passage that can help us answer this question. All right, different S. aureus strains preferentially infect different hosts and have different amino acid sequences at their hemoglobin binding domains. And that's in table 1, which I just talked about. In an experiment, different S. aureus strains were mixed with hemoglobin from macaque monkeys, and their binding ability was measured. And that's in Figure 1. And I'm not showing you Figure 1 yet, and I'll tell you why in just a few minutes. All right, the differences in amino acid sequences contributed to the differential binding abilities observed. So when you get an FRQ question, some of them have 3 or 4 different representations. They might have a diagram, they might have a table, a graph, they'll have all of them, and maybe 2 or 3 graphs. But each, when you answer part A, B, C, or D, it doesn't mean that you need to refer to all of them. There may be something that's very specific to your task verb, and you only need to go to that one. So that's why I just showed you this particular table. All right, so describe the process responsible for the different amino acid strains found on different hosts. So I'll give you a minute to write your response to that describe. Okay. Now, I'm not going to give you the answer to A just yet because now we have B. B, describe the trend illustrated. So in this graph, this kind of looks like what we just did just a few minutes ago. So on our X, we have S. aureus strains, 1, 2, 3, 4. And on our Y, we have percent hemoglobin binding. And so I'm going to give you just a moment, transfer the skill that you just did on a previous question to now this question. Okay, And then I'll bring up the answers to both. Okay, so your answer A should be similar to each strain is best adapted to a specific host species. And then B, strain 3 has the highest percent hemoglobin binding. Pretty easy, I think. Describe is going to be one of those that you need not leave that point behind, because all you need to do is talk about the characteristics of what you're seeing. Okay, so now for a debrief and summary of today. Hopefully, we were successful with the learning objectives for today. You should be able to describe the causes of natural selection. You should be able to explain how natural selection affects populations. The key takeaways: evolution is characterized by a change in the genetic makeup of a population over time and is supported by multiple lines of evidence. Natural selection is the mechanism of evolution. It was proposed by Charles Darwin in 1859. There are many examples of natural selection, and only some were presented here in this presentation. To describe, you should list the characteristics, patterns, or trends of a representation. Now, I want to talk about common errors and misunderstandings. In fact, there's a lot of that I'm going to talk about in the next several minutes. Hopefully we'll learn some things along the way, and then we'll get to the misunderstanding at the end of this conversation. So this was a question in the 2015 read. It was the first exam. It was my first year as a reader, and I had question number 1, and this was it. And it consumed all of us by the end of the week. So, and I'll tell you why at the end. Anyway, so let's look at our diagram. We have a mouse on the left that, well, we have 2 running wheels, and we have a mouse that's inactive and a mouse that's active. And then if we look underneath those mice, we're going to see what's called an actogram. And an actogram has the days on the y-axis, and then it has the time and hours on the x. Now, if you've never seen an actogram, my kids have never seen an actogram before this day either. And you're going to do that. There's going to be an exam where there is a new type of graph that you've never seen before, a new type of diagram, something you've never seen before. And your job is to interpret it. And once you have that skill set, you can transfer it from one diagram, table, or graph to another. So we can see that in the morning hours from 0 to 12, there is blank. And then in the evening hours from 12 to 24, there are these thick, dark bars. So we just look at that mouse. He's running on that running wheel, and it makes the dark black line. So we can see that the actogram has a black line with activity. All right, and that was at the evening hours. So that's what we should have pulled together from interpreting that diagram. All right, so let me read you part of the question. Many species have circadian rhythms that exhibit an approximately 24-hour cycle. Circadian rhythms are controlled by both genetics and environmental conditions, including light. Researchers investigated the effect of light on mouse behavior by using a running wheel with a motion sensor to record activity on actograms as shown in Figure 1. Okay, here's another part. So here we have another image, and it's another actogram, and we can see that, oh, there's a variable being introduced in this particular scenario eventually. Okay, so now we added time. We went over for 2 weeks. We have 14 days on our y-axis. We still have a 24-hour period, and we have most of our dark lines, our bold dark lines, are in the evening hours. So we can see our mouse is still running and being active in the dark. All right, so for the investigation, adult male mice were individually housed in cages in a soundproof room at 25 degrees Celsius. Each mouse was provided with adequate food, water, bedding material, and a running wheel. The mice were exposed to daily periods of 12 hours of light, and that will be represented with an uppercase L, and 12 hours of dark, which will be represented with an uppercase D. So L12, D12 means 12 hours in the light, 12 hours in the day. And they did this for 14 days, and they monitored their activity for those 14 days. Okay, now we have another actogram. And so now this one has extended to 30 days. And we have seen that we can see that there's a dark bar across. So it looks like there is going to be continuous darkness, and there's something cyclical maybe going on, but we can say that there are some activity going on in the morning hours and in the evening hours. Okay. After 14 days in L12, D12, the mice were placed in continuous darkness, DD, and their activity on the running wheel was recorded as before. The activity data under DD conditions are shown in figure 3. Okay, so let's take a moment to think about what we just saw. The mice are active when it's dark and not when it's light, okay? In nature, mice are potential prey for some predatory birds that hunt during the day. Describe 2 features of a model that represents how the predator-prey relationship between the birds and the mice may have resulted in the evolution of the observed activity pattern of the mice. So make sure you answer what the question's asking you. So you're going to describe it, which means list characteristics of this representation, and it wants 2 features of it. So there's going to be 2 parts. I don't want to tell you what they are until you think about it yourself, but there's a predator-prey relationship. We have that. And what that has to do with how this activity pattern has evolved. So we have a bird eating a mouse, and we have birds eating mice, and then we have activity during the dark. So I want you to take a few minutes and write 2 features, describe 2 features of what would have happened along the way to make this happen. Okay? Okay, let's see what the scoring guidelines say. Okay, so here's the scoring guidelines, and there are the 2 features that they wanted you to talk about were the selective advantage and the reproductive success. So if you're a mouse, or if you're a population of mice and you are awake and active during the day, the birds are going to eat you, okay? So those mice active during the day will be selected against. If you are a population of mice that are active at night, then the birds will not be able to see you. And so you will be selected for if you are active at night. So that's a selective advantage. So if you are dead during the light hours, you're not reproducing. But if you are living because you had the selective advantage in the nighttime hours, then you are the ones that are going to reproduce, and you'll have generations of offspring following, and you will be the fit in that environment. Okay, so that's where they were heading with that question. So we went over a lot. We reviewed again some natural selection, but my point to this was, I want you to read what you wrote on that piece of paper, and I want you to look and see if you said anything that looks like the mouse evolved, like this mouse who decided, like, I better stay up at night, okay, or I'm going to be eaten, and I just need to change my routine until I'm eaten. If the mouse evolved, that's what you're kind of saying. So that's called Lamarckian, and we don't accept that when you write about populations. You need to say the mouse population evolved to be, they were selected for, and then they had reproductive success. Okay. All right. Next is some AP exam practice. You're not going to do this right this minute. This is going to be your homework, even though if you're all home now, I'm not really sure what homework is called. Anyway, if somebody has an idea, you can send that to our way on Instagram or Twitter or whatever it is that you want to talk about this on. Anyway, so I'm not going to, you know what I want you to do. I want you to go to look at that diagram and figure it out. And then I want you to go find the task verbs. What do I need to do? Read the passage and then answer this question. And then tomorrow night, well, I won't say tomorrow night for some of you who will be watching it another time, but on the next lesson, I'll be starting off with this particular question as our warm up. So you can, after this says, I'll give you some time after, well, the video will be saved and then you will be able to rewatch it to get all the details of this question again. And thanks for watching. We hope you'll join us again. Be sure to complete the practice question and we'll review the answer in the next lesson.
Thank you. Bye-bye.